So this is the handout staining identification. And so the first stains that we did and we learned about that you need to know for your exam is simple stains versus negative stains. So I have an example of each. What charge do bacterial cells have, positive or negative? Negative. And this is why they stain differently with the different dyes that we use, because the different dyes we use have different charges. So in this above picture, we used a basic dye. What charge do basic dyes have? They're positively charged. So in this case, we stain the cell because the cells are negative, the stain is positive, opposites attract. So you notice the cells themselves have taken up the dye. What dye do you think was used to stain this slide? It's blue. Do you guys remember the blue stain we used? Methylene blue. That's a basic dye. Some other ones that we've used and will continue to use are crystal violet, that purple dye, right? And saffronin, which is red, right? But remember when we employed it in the gram stain, they didn't hold on to a lot of the red dye and they looked what color? A lighter shade of red is pink. So again, positively charged, right? Opposites attract. So if cell's negatively charged, dye is positive, dye goes into the cells. The next one is an acidic dye. Anyone know which acidic dye was used to make this slide? Has this colorization to it. Had a grayish background. Starts with an N. Nigrazine. We also use Congo red, which made the background red in color, right? What charge do these acidic dyes have? They're negative, and we know the cells are negative, so what happens here? You get repulsion, right? So what gets stained? The background, not the cells, because like charges repel each other. So also in this picture you can see a particular arrangement where you've got 4 cells together in that little square conformation. What is that called? Tetrad. And each individual cell is a little sphere, and what's the term for that? Coccus if you're talking about one, cocci if you're talking about all of them. So gram stain, very important procedure, a differential procedure, right? So we're going to end up with 2 different results. Remember, gram negative and gram positive, we're talking about a result, right? When we say gram positive, it came out what color as a result? Purple. Gram negative, pink. So for this stain, the first dye we use, right, the primary stain is crystal violet. Then, after we rinse with water, what's the next thing we apply? Iodine, right? In your book it said grams reagent. It's iodine. Again, rinse, and then we decolorize. And what do we use to decolorize? An alcohol, a specific alcohol, ethanol. I remember this one we want to do drop-wise until it just starts to lose its dark color purple to those drops. You don't want to do it till it's clear. You could over-decolorize. Remove it even from your gram positives. Now, in order to see our gram negatives, we got a counter stain. What's the counter stain? Saffronin. It's going to make them appear pink. We blot dry. Do not rub. And remember, always with bacteria, we're going to go all the way to oil immersion, right? You're going to live the rest of this course under oil immersion, right? Whenever you go to look at anything, it's going to be under oil. All the way up to 1,000 times magnified, right? So here we have some tetrads. We have 2 different results for the gram stain. So these ones are what color? Purple. What dye made them that color? The crystal violet. And they did not decolorize, so they stayed purple, and so their result is gram positive. These other cells are what color? Pink. What made them in this color? Saffronin. And they're gram negative. What's the shape for both of these cells? Cocci, right? And definitely tetrads here, they've got that labeled. Here, what are you seeing mostly? I see some clustering, I see some tetrads, right? But I see more pairs. Don't you see that? Pair, pair. So that would probably be diplo. Right? If it was clustered, you'd see more than just one cluster. If it was tetrad, you'd see more than one, right? I see one here, maybe two there. Mostly I'm seeing pairs. You see that? So that's how you would determine arrangement for that one. So what does gram-positive and gram-negative tell us? Well, it tells us one's purple and the other one's pink. But why are they purple and why are they pink, right? And it has to do with their cell wall structure, What substance is this cell wall made out of? Peptidoglycan. For gram-positive cells, how many membranes do they have? Just one, right? Just the normal cell membrane that all cells have. This, unfortunately, has multiple names, right? Plasma membrane, cytoplasmic membrane. These all mean the same thing. I typically just say cell membrane. It's pretty self-explanatory. But understand that all these terms are the exact same membrane they're talking about, right? Whenever you see that. The peptidoglycan layer for these cells is thin or thick. It's thick. And remember, this thick layer is why they stay gram-positive. It traps in the crystal violet iodine complex. And remember, crystal violet goes in, iodine goes in, they bond, they become a bigger molecule. And it gets trapped in. Unlike gram-negatives, they tend to have how many membranes? 2. And again, the normal membrane that all cells have to have, and what do we call this one? Outer membrane. That's easy, right? Because it's the outer one. And so again, remember the cell membrane has multiple names, but thank goodness the outer membrane just has one, and it's really good clue as to where it's located. Now, if they've got peptidoglycan, for them it tends to be thick or thin. Thin, right? So notice sandwiched between the two membranes, a thin layer. And this isn't enough to trap in the crystal violet iodine, and we also have this problem with this other membrane. What happens to membranes when they're exposed to ethanol? They're fatty, right? They're phospholipids. They don't shrink. They dissolve, right? So you get these big pores, right, and the crystal violet iodine gets washed out. And then they become colorless. So if you don't do the last step, which is staining with safranin, you're not going to see them. Right? We talked about all the problems that could go wrong if you do any one of the steps, if you skip them or don't do them in the correct order. Right? The different consequences you could be dealing with. The other stain we did last week is a capsule stain. And so which picture actually has a capsule? A or B? A. And what indicates that for you? The halo, right? So the unstained capsule, which is going to be outside the cell. This is outside the cell membrane, outside the cell wall. Right? This would be the outermost covering if the cell has it. In this picture, you don't see any halo, right? And the reason why we know these are capsule stains because the background is stained, right? And so is the cell, right? Both have to be stained in order to visualize the capsule if it's present, right? So why do we need to stain the cell in the background? Because remember, if we did any one of those stains, if we just did a simple stain, we just stained the cell. And then that clear that unstained capsule would blend in with the clear background. If we just stain the background, all we're going to see is a clear cell. And we can't, again, distinguish the cell from the capsule if present. You got to stain both. And because of that, remember, we employ different stains. Negative stain for the background, Congo red. And then we use the Menville's reagent, which is basic in nature. It's going to stain the cell. And it even changes the color of the background, right? We end up with a red slide that ends up being a kind of grayish slide. There are two functionalities that we talked about that capsules provide for organisms that are beneficial to them. Protection, and what do we mean by protection? Invisibility inside of our bodies. It helps it avoid that process of phagocytosis that we watched, right? So very simply, you can say protection, right? That's a one-word description of it, sufficient, right? And that's what we mean by it, though, is that, you know, when they get into our body, it's like they're wearing a disguise, right? So these are pathogenic organisms. One of the ones we looked at was Klebsiella pneumoniae, right? It has a capsule. One of the reasons why it can cause pneumonia so well is because of that capsule allowing it to hide from our immune system and grow rapidly and create destruction in the lungs before we can really get a handle on it. The other is related to, again, to our bodies. What does that capsule allow it to do? Attach, right? It allows it to attach or adhere. So again, we talked about these guys inside of our mouth, right? And that allows them to attach to our teeth. So attachment and protection are the two main functions of a capsule. So a lot of pathogenic bacteria have capsules because it's beneficial to them, right? To be able to attach to us and protect them against our body's defenses. One of the stains that you're going to do today, you can thank Raina because she helped me prepare our smears for you guys today. So the smear is already done just like the gram stain. You're just going to need to stain it. So while your other stain smear that you guys are going to prepare is drying, you'll be able to start staining these ones. And so we have two organisms smeared on the same slide for you. We have an acid-fast organism and a non-acid-fast. And this is a differential stain, just like gram stain. So we're going to end up with two different colors, which tell you two different things about the organisms. So the dye we're going to use today is a reddish purple dye, sometimes referred to as a fuchsia color. The dye is called carbofusion. And there are two different methods that are widely used of this staining procedure. So they have a little bit of differences in them. We're going to use the Nelson, Nelson, commonly abbreviated ZN. Good news, you don't have to spell his name on the test, but you have to recognize it. It's actually two people's names, obviously, put together. Came up with this procedure. We're going to be using, it's a little bit less concentrated form of this dye than the other method that we use. And there's several reasons why we use this one instead of the other one. This one tends to come out better in the lab setting that we are in. So these are what we call acid-fast. Instead of saying acid-fast positive, we typically just say acid-fast. But acid-fast positive is another way of identifying that they do stain with this primary stain in this staining procedure, which is carbofusion. We'll get into the particulars of the stain when we get to today's lecture. The other color, what color does it kind of look like to you guys? Kind of bluish, gray, right? Even some people say it looks greenish, blue, like an aquamarine color. The reason for that is the dye that we use for our procedure is methylene blue. Other procedures, sometimes they'll use brilliant green. Again, they're going to look this lighter color. They're definitely not going to be the acid-fast color of this purplish pink fuchsia color. So these are what we call non-acid fast or acid fast negative. They did not retain the dye. They did get decolorized. And the decolorization we use in this staining procedure is partly where the staining procedure gets its name. The decolorizer we use is acid alcohol. Actually, it contains hydrochloric acid. So really strong acid is the acid you produce in your stomach. So despite that, right, these guys stain so well with the carbofusion that even with that really strong decolorizer, they don't decolorize. So they're acid fast, right? They stay stained. Fast meaning staying, right? Despite the use of acid. where the other guys, of course, decolorize, and we stain them with our counter stain, right? That's contrasting to our primary stain. The next stain that you guys are going to do today is the Endospore stain. And this one, too, has many different methods out there. The method we're going to employ is the Schaefer-Fulton method. In this method, the endospores, what color would you say these endospores are? They're kind of green, right? I'm finding sometimes they look kind of bluish to me. So again, like a greenish blue. What dye made them this color? What's the dye we're going to use today? Anyone know? Malachite green, right? And you should have this in your procedure for today. These are the bottles that are the most stained and most difficult to read the name on it. So it's a greenish looking bottle. You kind of can't miss it. So on today's quiz too, depending on if you were able to catch one like they did here, do you see how the spore is still inside the mother cell? Where would that location be, terminology-wise? Subterminal. Good job, y'all. So it's not smack in the middle, right? And it's not all the way at the end. Smack in the middle would be central. All the way at the end, terminal. So this one's kind of in between central and terminal, so it's subterminal. Now, if we get lucky today, you might catch some in that state, where the cell itself has not deteriorated. Most of the time we're going to see spores all by themselves, what we call free spores. And again, the proper terminology would be endospores. And these are only formed by bacteria, and only by ones that have this ability, and only when they're in harsh conditions. So the harsh conditions we employ in this lab is we starve them. So I started these cultures last week. So we should definitely have spore formers. We should have spores in these for you guys to look at. Now, whether we'll actually catch any before the cell deteriorated or not, we might get lucky. So I do want you to scan around and try and find one like that if you can. The other thing is, you'll see they're teeny tiny under the microscope. You guys have already looked at bacteria, right? Being able to actually get one where it's like next to impossible mission, right? So under the microscope, I won't ask you to determine location of a spore, right? You'll just be able, you need to be able to identify if you are in fact seeing spores, right? And the cells themselves in this procedure will get stained red. So in this picture, do you see spores? No, you're just seeing the cells, right? Because if you did see the spores, they would be, right, these greenish structures, whether inside the cells or outside. So what type of conditions, what did I say we employ in lab? Starvation, nutrient depletion. But other things, other harsh conditions. So can you guys give me an example of a harsh condition that might cause an organism to go into this dormant state? Temperature. What's something else an organism might need to live? Moisture, water. Excellent. Anything else? Food is nutrients. Anything in the air, maybe? Oxygen, carbon dioxide, different gases, concentrations can cause them to form endospores. So if they don't have what they need, they're going to go into this dormant cell state. And what's the most precious thing inside that cell they're going to preserve? Their DNA, right? DNA is the most precious thing. That's what they're preserving inside that cell. So based on what we know about the stains and the colors that we should see in the different structures, underneath the microscope, you guys have to be able to determine what type of stain you're looking at, right? So when you look at this one, what structure are you seeing, what colors are you seeing that would indicate what type of stain may have been done? Endospores, because what do we see? Green endospores and red cells, right? So endospore stain. This next picture, what do you see? You see a dark background, and the cells inside are colored too. What color are they? They're purple. So we stain the cell and we stain the background. Why would we do that? We're usually looking for a capsule. But do these guys have a capsule? No, because there's no clear space between the cell and the background. So this is a capsule stain, but it is negative for a capsule. The next one, what colors do you see? Well, first of all, how many colors do you see? 2. So it's probably differential stain, right? But what colors do you see that'll help us determine which differential stain? We see that purplish, reddish color, right? And kind of bluish other cells. Another key characteristic you'll notice about these cells is they tend to clump together. But when you look, see how they're rods? Bacillus? So anyone guess? This is acid fast. This is one of the ones you'll do today. And hopefully it'll look like that. The next one, again, 2 different colors. What 2 colors? Purple and pink. So that's easy, right? Gram stain. Purple is positive, pink is negative. Okay, the last stain, what do we see? Background only, right? Stain, the cells are not. So this is a negative stain. How do you tell the difference between a negative stain and a capsule stain? In the capsule stain, the cells should be colored as well. So the quizzes that I have online, the bonus in that, go over this, except it tells you what stain you're looking at, and it's just asking you results. I guess I should make one where you have to figure out what the stain is. I don't think that's that hard, though, right? Y'all can do it. Usually my questions kind of clue you into as to what you're looking at. Okay, so go over morphology, shape, and arrangement. So what do we see in this first picture? What's the term for this shape? Cox eye, referring to all of them, just one? Coxus. What's this term for this shape and arrangement, where they're in pairs? Diplococcus, if you're talking about one pair. Diplococci, if you're talking about all the pairs, right? What's this arrangement that we see here? Tetrads. This is a chain of cocci, so this is Streptococcus. Remember, it causes strep throat. Wouldn't want to swallow a chain when you have strep throat. It would hurt like hell. The next one is a packet of 4 cells, right, in a cube arrangement, these cocci. Sarcinia. And then the clustering is Staphylo, not to be confused with strepto, right? What's the individual shape of these rod shape? What's the term we use? Bacillus, or bacilli if we're talking about all of them. And then, of course, in pairs is diplo, but this is a bacillus, so we say diplobacillus. Rods can also form chains here, so what's our prefix? Strepto, and they're rods, so it's streptobacillus. These are rods that have a little bit of a curve to them. Even in this picture, they also have a flagella. Vibrio. This one is a rod with multiple curves. Spirillum, or spirilia if there was more than one. And then this is all the same organism in this picture, but no characteristic shape. What is this phenomenon referred to as? Pleomorphism, or being pleomorphic. These guys look like squiggly lines under the microscope. 3-dimensionally, they would be helical in shape. These are your spirochetes. And these kind of corkscrew through the environment. These are highly pathogenic and hard to grow, so we don't get to look at any of these under the microscope here. So what should you study for your midterm? All the exercises we've covered so far. What are good examples of questions? Your quizzes, right? Which have been graded and given back to you, except for today's, which I'll do during class. You should also know all the terms you were assigned for homework. This includes being able to spell them, sorry. there will be lots of slides that you will have to identify. You'll have to identify both prokaryotes as well as bacterial stains, which we just reviewed here. Prokaryotes, I think we've kind of pounded that one in pretty good, and again, I've got that extra quiz for you guys for learning the names and identifying as well on Blackboard. For eukaryotes, you should be able to identify characteristic structures and the names of the organisms, right? So here's some examples, right? You should know all the questions associated with the organisms from that homework handout and the lecture we went over. So for instance, if I had Aspergillus, which is a mold, do you guys remember what its asexual spores look like? It had that ball structure, right? It's almost like a dandelion, right? So I could have the pointer on those little balls, right? And that would be the asexual spores. The question that was in your homework about them was what commercial compound do they produce? They produce citric acid, right? And they can also be used with soy products to produce soy paste and soy sauce. Any one of those would be acceptable. I don't expect you to put all three. Right. Remember at least one of them. Alright, so make sure you go over those. That bonus quiz you guys took helped you out with that as well. You need to know all the steps to all the staining procedures and why we do what we do. Right? So what's happening in the first step of the gram stain. Right? We're using crystal violet, which is a basic dye. Right? It's positively charged, goes into the negatively charged cells. Then iodine goes in and combines with the crystal violet. And then it gets trapped in when we decolorize, because what do gram-positives have? That thick cell wall, right? I was looking for those models and I walked away. I took them. Okay. Again, with the capsule stain, why do we do the negative stain portion? To stain the background. And then the next portion we're staining the cells, right? And the capsules don't stain, right? That's why they look clear. You need to be able to identify what staining procedure. Again, we went over that in this assignment. You need to identify if you see things like endospores in capsules. You should be able to distinguish gram-positive from gram-negative. Same thing with acid-fast and non-acid-fast. If I put a slide up there, right, and say, what are you looking at, you know, acid-fast or non-acid-fast cells, right, be able to identify. Don't forget to use... We used different methods. So we're using the ZN method for acid-fast and Schaefer-Fulton for endospore. It's very important to know what method you're using, because that also is why you see the colors that you see, especially for the endospore stain. There's a couple of different methods out there, and they use different stains, right, in different procedures, and so therefore you get different colors. It might throw some people off. So there are certain characteristics about the organisms that you need to know. And some of them we haven't worked with, we'll work with today. And so sometimes we abbreviate their names, right? Sometimes we abbreviate the genus name. So that's what we're showing here, right? So Mycobacterium smegmatis is one of the organisms you'll work with today. This one is rod-shaped, and it tends to cluster. This is our acid-fast organism. So its acid-fast reaction is acid-fast or positive. Escherichia coli, or E. coli for short, you guys remember the shape? Rod, bacillus. Does it form a characteristic arrangement? Do they stay next to each other in chains or pairs? No, they're what we call single. Right? They're all by themselves. They don't stay next to each other. I used to believe at one time it's because they're modal, but a really cool video I want to show you guys today. I'll have to go grab it to my office. The kids that I've been working with in the mornings, one of the organisms you guys are going to work with today, we videotaped moving under the microscope, and they're bacillus, they stay together in chains. And so you see this really long chain of bacilli swimming, and it almost looks like a worm, right? So even ones that are modal will sometimes stay together and move together. So it's really cool. But E. coli is not one of them. He's kind of solitary. So when we stain this one, it should come out pink in the gram stain, so it would be gram negative. And then acid fast one of the things you'll learn today is only one genus of bacteria. Now notice I'm referring to bacteria. Other things you could stain with this procedure, and some of them would come out acid-fast. But as far as bacteria goes, only the genus Mycobacteria are acid-fast. So having said that, can E. coli be acid-fast? No. It's going to be negative. If we were to stain it using the acid-fast procedure, it would not come out that carbofusion color, that pinkish purple color. It would be whatever the counter stain we were using, like a bluish gray. Micrococcus luteus, what was its shape? Individual cell shape. Cocci. And it forms a characteristic arrangement, right? Of 4 cells together in a square. It's a tetrad. Its gram stain reaction would be purple, so it is gram positive. Acid fast, what would it be? Non-acid fast or negative, right? Because it's not a mycobacteria. Repeat after me. It's not a mycobacteria. So therefore it'll be negative. Staphylococcus epididymis. Shape. Cocci has it in its name, right? Arrangement's in its name too. It's going to be clustering. Gram reaction, it would come out purple, so it's gram positive. Acid fast, what would it come out? Non-acid fast or negative. Klebsiella pneumoniae. Anyone remember its shape? It's rod, bacillus. Does it have an arrangement? No, these guys are singular, all by themselves. If we were to gram stain them, they'd come out pink, so they're gram negative. What special structure did we do a special stain to be able to visualize for these guys? The capsule. They have a nice thick capsule. If we were to do acid fast staining, they'd come out non-acid fast. Today, we'll use 3 different bacilli. Bacillus cereus, Bacillus megaterium, and Bacillus subtilis. By their genus name, what shape are they? They're rod-shaped, right? They're bacillus-shaped. I think all these guys form chains. So you could also say they're streptobacillus. And if you stain them, they would all be purple, so they're gram positive in the gram stain. The reason why we're using them today, anyone know what special structure they'll have? They will have endospores. Lots of bacillus have endospores. Of course, we're not going to work with the most famous one, Bacillus anthracis, because it causes anthrax. Right? So these are non-pathogenic bacilli that form endospores, especially when you starve them, as I have done. So each group will get a different bacillus, so make sure you circle on your data sheet today which one your group is looking at, right? And that's how we'll keep track of what organisms you guys are doing. So if we were to stain using the acid-fast stain, these guys would be negative. Now, under the microscope, I can't just put a slide and have you tell me what organism it is, right? But if I asked you, like I did on today's quiz, right, if I said E. coli, you should be able to tell me what? It's rod-shaped, singular in arrangement. What else? It's gram-negative and non-acid fast. Right? But I would not be able to show you a slide and say, tell me which one it is. You can't do that. And you guys will definitely know that after midterm when I give you an organism, right, that of course I know what it is, but you don't. When you're given the unknowns, you're going to run all these tests and stains to determine what it is, right? So you're going to take into account a whole bunch of information to be able to figure out which one you have. Okay? Make sense? At least that part will, hopefully, after midterm. It's fun. Students like it's like a mystery, right? Love trying to figure out what you got. Oh, so I even had the answers there. I forgot I had them there. So the other thing is, remember, with the gram-negatives, it's that thin cell wall made out of peptidoglycan, if they have it at all, is why they're going to come, and they have that outer membrane, why they're going to be negative. So that was the only thing that I think that's the last slide. At least it should be. Yes. So does anybody have any questions at this point about midterm? All right, so study your homeworks, study your quizzes, study this handout, study the extra quiz I put on Blackboard, which I'm going to go to right now. And in fact, we'll do the bonus one together, but you can do it again for your bonus points. So let me go to our main homepage. So after you've studied, you definitely, and before the test, you want to do these. So the bonus and the quiz file is just like the bonus, is just longer. So notice it says here, looking at this picture, what's the shape and arrangement of these cells? So individual cells, you can kind of see right here, so they would be rod shaped, which we call bacillus. Do you see any characteristic arrangement? So for the bacillus, they're not single, not all by themselves, they're clumping together. Right? This is very common for acid-fast bacilli, AFBs, not acid-fast bacteria. Hope you didn't miss that on the quiz today, right? If you had your homework in front of you, you shouldn't have missed it, right? So it's a common abbreviation for these guys, and they tend to clump together. So this is them again, but gram stain. So what color do you see? So they're gram positive. Acid-fast. They're positive, right? It's that purplish red color. And they're clumping, right? And they're bacillus shape. Those are all clues. Spore stain. Negative, because we're not seeing the green spores anywhere. Just the cells. Capsule stain. Negative, because no halo. So then you hit save and submit. You hit OK when you're done. And it will tell you, right, whether you got it right or not, right, and remind you, right, under the feedback, why things are what they are. So we'll do the same for this quiz, right? This is a good practice, and then take the next one, which is going to have 2 questions of each for staining identification. Okay, so today's lecture, which is on acid-fast and endospore. And I might do this on 2 separate recordings, so let me